Hello, I'd like to present to you my project titled The Enhancement of Doxorubicin-Induced Apoptosis in U2OS cells in the presence of hyperthermia. Now, I'd like to note that this is an individual study, a hypothesis-based study, and also falls under the category of molecular and cellular biology. Now, initially, I had received the idea or inspiration for this project from my mentor, who had instructed me to look at the effects of microwaves on osteosarcoma, or a type of bone cancer. Now, microwaves have been shown to be adjuvant therapies, much like heat, cold, and radiation. However, when looking at the effects of microwaves and going through the established literature on PubMed and Google Scholar, I have noted that it seemed to be a relatively well-established issue. Now, after seeing the thorough investigations, I decided to shift the focus of my research investigation to hyperthermia, or heat. Now, heat is also an adjuvant therapy that is used in conjunction with current treatment methods to increase the effectiveness of those current methods. Now, chemotherapeutic drugs are one such example. Doxorubicin, as used here, is a drug within the class of anthracyclines, meaning that it inserts itself into DNA's bases or intercalation and induces double-stranded breaks that prevent the proper replication of DNA. Now, this can be detrimental to a tumor's proliferation. Now, doxorubicin has many adverse side effects. Those include cardiotoxicity and also reduced immune system function, which can both be very dangerous for already immunocompromised patients. But osteosarcoma tends to occur in younger individuals due to areas of active bone growth during those years, usually 10 to 20 years old. And osteosarcoma often involves limb salvage surgery, but also chemotherapeutic drugs, such as doxorubicin. Doxorubicin is one such drug used to treat osteosarcoma. Now, I decided to shift the idea of my project by changing the keywords during my literature review. And while looking at that, I had found that the effects of hyperthermia and doxorubicin on osteosarcoma, while well documented, had not been investigated simultaneously. While they were shown to be effective and have a negative impact on the osteosarcoma cells individually, their use in combination had not been investigated, and so that was how I decided to modify my previous idea. Now, when looking at my topic, it seemed clear that I had to design an experiment to test whether these treatments would be effective and more effective than when either of them were used individually. Now, I decided to go about answering my research question, which was, does hyperthermia increase the effects of doxorubicin on the U2OS cell line? Now, I went about answering this question by devising an experimental design and establishing 4 experimental groups. Now, I cultured the U2OS cells, and based on their logarithmic growth, I decided to use the cells that were able to be treated. Now, the cells were also underwent pre-experimental trials in order to determine the concentration of the doxorubicin required. Now, 100 nanomolar of doxorubicin was used for the doxorubicin group, and the 42 degrees Celsius for 20 minutes was the hyperthermia group. Now, there was also the first group, which was the normal thermic control group. Now, the results of the other 3 groups were compared to the first group. The first group was, again, the normal thermic control group. The second was the doxorubicin-only group, or 100 nanomolar doxorubicin being injected into the cell culture medium, and then the cells were incubated and for 72 hours at a temperature of 37 degrees Celsius. The third group was the hyperthermia-only group, in which the cells were placed in small Eppendorf tubes and submerged in a constant water bath and maintained at a temperature for 20 minutes. Now, group 4 was the combination group, in which both treatments of groups 3 and 2 were applied simultaneously. the cells were again cultured with the same conditions and incubated. Now, when looking at the results or analyzing the data of these, I was able to learn that the average cell survival rate, which was looked at in order to determine the cytotoxic effect, and the fluorescent microscopic analysis had shown supportive results that seem to reject the null hypothesis. Now, hyperthermia has been shown to somewhat limit the effects of, tumor cells proliferation. Now, group 1 had the average survival rate of 100%. Group 2 had a mean survival rate of 85.4%. Group 3 had the average survival rate of 96%. And Group 4 showed a significantly reduced survival rate of 69.4%. And thus, the combination group was shown to have a significantly inhibitory effect on the tumor U2OS cells proliferation. Now, when looking at the fluorescent microscopic analysis, the cells were treated with MitoTracker, and they were stained and became visible under the fluorescent microscope. Group A was Group 1. Now, Figure A shows the cells that underwent no treatment. Group 2 is Figure B, which shows cells that underwent the doxorubicin-only treatment. Now, the cells exhibit significantly fragmented mitochondria in that there are no networks and they are sparsely spread out. Group C, or Group 3 in Figure C, shows the hyperthermia-only treatment, in which the cells had shown to be virtually invisible mitochondria. Now, MitoTracker only stains the mitochondria and is visible if there is an active proton gradient established. Group 4 showed virtually no mitochondria active, and thus led me to conclude that the mitochondrial membrane potential had been altered. Now, when looking at the science behind these results, I noticed that after determining that that the cell survival rates were statistically significant with a p-value of less than 0.05 from the analysis of variance test, when looking at the actual science or molecular biology behind it, there are numerous mechanisms that seem to explain why hyperthermia would have such an effect. Now, as I mentioned, there was a inhibitory effect on the mitochondria in groups 3 and 4. And these were the hyperthermia treatment groups. And so hyperthermia has been shown to have a strongly inhibitory effect on osteosarcoma. However, its role as a chemosensitizer with doxorubicin when used in conjunction had not been investigated. Now, adjuvant therapies or conjunction therapies have been studied and used clinically to increase the effectiveness of the current treatment regimens. Doxorubicin's side effects, which as I mentioned were harmful, could be lessened by lessening the dosage of the chemotherapeutic drugs and thus lessening the adverse side effects. Now, mechanisms that could potentially explain how hyperthermia acts upon these cells include cellular redox status being altered or reduction oxidation status, and also the ability of the cell to produce its energy source or ATP within the mitochondria. And final mechanism that could account for this would be effects of the DNA damage repair mechanisms in the nucleus, or the homologous base repair mechanisms that could be affected, or certain enzymes that would otherwise be able to replace mutated DNA bases. Now, it is also known that the preferred temperature range for hyperthermia is roughly 42 to 43 degrees Celsius for a duration of 20 minutes to an hour. And it is these conditions that have been shown to be beneficial to other cells, as in they don't harm healthy cells while also maximizing the effectiveness onto tumor cells. Now, in conclusion, hyperthermia was shown to increase the effects of doxorubicin on the U2OS cell line, and in terms of doxorubicin's apoptotic effect. Now, I was personally very invested in this topic, as not only had I known somebody who had been afflicted with cancer, but I was also very interested in the field of oncology. Oncology I felt was very interesting as drug resistance was becoming an increasingly discussed issue within the scientific field, and I wanted to investigate it further. Now, drugs that have been shown to develop resistance were a major obstacle for the discipline of medicine, and so I decided to investigate that with the help of my mentor. I also learned many other things during this investigation and had many valuable experiences. I took away the fact that I learned much about the scientific method and was able to gain a thorough understanding of it. I was able to develop an experiment by reviewing the literature to determine what was the gap in the current body of knowledge, as well as continuing to develop and create an experiment that would target that body of knowledge gap. I also learned how to analyze the data in terms of tests of statistical significance, and finally learn how to draw a conclusion from that. I was also able to learn how to personally take away the fact that there were many concepts in cellular biology that I felt were very interesting to me, such as the fact that the mechanism by which doxorubicin acts. I was also able to take away one final thought, which was how to work in a laboratory environment and also use basic cell culture procedures, as well as learn how to apply drugs in vitro. Thank you for your time.